I'm covering a topic today on miracles, and these are biochemical miracles. And the title of my talk is Miracles at Your Feet. Now, most people who are poets or whatever know this statement by Joyce Kilmer, a poet. Poems are made by fools like me, but only God can make a tree. And the whole purpose of this is to say how complicated making a tree is. So this is directed toward people in our population, presumably educated people, who have good knowledge of everything except for biochemistry. So I'm filling in this part of their education in a very simplistic, understandable way. For non-biochemists, I would like to set the stage that in my lifetime, between the 1933 and now, there's been a vast amount of information accumulated in biochemistry, and it's been very intense. And I'll be referring to this chart a little bit later, but this just shows you the complexity of what is occurring within a living cell. And through the years, the scientists have tried to figure out how the living cell became possible. And when the Earth was just developing, of course, all there was this was all the elements of the periodic chart. And so they were incorporated within the Earth's crust. And so people have tried to see how various harsh chemical and physical conditions can occur that would make components of a living cell. And Stanley Miller tried to do this in the 50s, and it was very popularly accepted. He was able to, using forces and electricity and whatever else, accumulate particles of things which were amino acids or bases, which are components of the nucleic acids. So I'm saying that the most people that philosophize by things that occur on the Earth, really to be solid in their interpretations, should know really what has accumulated to produce a living cell. And to have an example of this, a few years ago I was fortunate to witness the action of a showman, I guess is how it's pronounced, in Ecuador. He had the beneficial actions that in his own way would help his clientele, but his limitations were limited to his knowledge and experience. And so he was able to, with his batter and stuff that he made, feathers and whatever, to give them some assurance he knew something about medicine. One of my earlier experiences was with the fungus psilocybin mexicana. The boss I had at that time wanted to know if the alkaloids of this fungus, psilocybin and psilocyan, were synthesized by the same pathway as the tryptophan is in this pathway here. So we got the fungus, but there had already been one man in an Indian tribe that knew of its properties, and he used the psychedelic mushroom itself, which contained these compounds, as a tool to predict the future, cure the sick, or anything his mind could develop after consumption. My corresponding point is that many men interpret Earth's experience based on what could be seen or surmised. And as I mentioned through the days, what we have now in the knowledge is tremendously more than people in biblical times, and even in times in my earliest days when they had Dust Bowl and whatever else taking over the country. An alternate explanation is that things did not accumulate by self-assembly, but that it was a supernatural power, of course God, who in 7 steps made all these things possible in all the life forms that we see. If that's the case, then we don't have to worry about this other thing, but what happens in terms of self-assembly. But I would like to show you how complex things are if it was by self-assembly, what would have to happen. If you reverse engineer anything, you need to find out how it works. You start with the simplest form and then try and analyze that and work the complex. However, in biology, the simplest organism currently known to science is still remarkably complex. We know everything living has assembled with the same types of building blocks. All the biological organisms have DNA, protein, carbohydrate, fat, enzymes, and small molecules like sugar, amino acids, sometimes vitamins. Now there's no half-constructed parts or pieces of cell that you can start with. Every little insect, mite, bacteria has the same basic mechanism that we see here on this chart. And this chart was actually assembled in 1974, so there are things that are well beyond that are known. The other thing is, no matter whether the organism is big or small, it still has control of these items with 10,000 components of intermediary metabolism that must come through activation of genes. And not only that, this DNA which contains the genes must be divided into and replicated so that when a cell divides, you can put the same information in the daughter cells and maintain it. Now DNA is the first miracle, really, that we have put our hands on. It's a double-stranded strip of 4 chemical compounds, bases, which are strung together in a long length, and specific sequences of the information can make an organism to precision. However, DNA by itself in the world without cellular life can do nothing. With an enzyme and some molecular help from a short piece of DNA message, you could transfer this information to RNA if you have an intact cell. Still, without the cellular help, RNA can do nothing at all. In machinery in the living organism, this information can code for an enzyme. Again, the enzyme by itself can do nothing at all. So you have to have an intact cell, and in the presence of some small molecules like substrate, the enzyme can then catalyze a reaction. And what you see here is a bunch of catalyzed reactions. Now the simplest known living cell has 10,000 individual strips of DNA, which are essentially genes within the organism's genome, that are capable of doing the miracle of events described above, each carrying out a function required by the living cell. And for the function in the cell to be carried out, it has to rely on an organism set up in the cell to convert the message to a protein. And there's things called ribosomes inside the cell, and even the ribosomes themselves are made up of proteins, so it's you can't have one without the other. So these are very, very complex systems, and there's no shortcut. You have to have the whole thing here to get the cell in a living state. There has been a cataloging of all the information, of all the DNA that is contained within all the organisms you can think of, from bacteria to fungus to wheat to peas, or whatever. Peas is what I work with. So there are things called Gen Bank where you can go find a certain gene and then see what these genes look like compared to other genes in other organisms. Now, why is all this information important for somebody to philosophize about? My main concern is that the typical man in the street, when he hears something in the news like water is found on Mars or some planet, that there's always almost immediate speculation that there's life on Mars, or has been, or should be, or will be. However, water is only one component that the planet would need to have life. And all these other things must be there accordingly. And in most of the planets, all we have are elements of the periodic chart. Okay, now we have some sort of an idea of the complexity of a living cell. It is a living cell that must be there before we can even think about starting the evolution that people have talked about. And evolution was first featured by Charles Darwin in this book, Origin of Species. So his interpretation was it was a series of mutations that caused organisms to change or to move or to improve or develop certain traits. And so he went to the Galapagos Islands where he saw these diverse types of microorganisms and fungi and sea-going animals and even land items. And crops, he also was interested in crops. So how does mutation work? Well, mutation is primarily an alteration of the DNA molecule. So if you can mutate the DNA, you can make it produce a protein that is a little bit different than it did before. And evolution actually has not really changed the functional proteins that much. And I just want to point out that what the plant geneticists have figured out, I work with peas, and peas have a DNA molecule that's 1 meter long, and there's one of these in each of the cells of the tissue. So if you see where this DNA for one cell is located, it's in an area that is small that you have to look at the microscope to see it. And then within the cell, it's in the nucleus, which is even smaller. It's 10 microns across. So everything that the DNA does has to be done in that little tight space. So if you were trying to untangle wires or whatever else, knitting yarn or whatever, and get it separated into two parts, it would be a real struggle. But one of the miracles that the biologically developed cell has figured out is that it has enzymes that, first of all, replicate the DNA. It's got other enzymes which take one strand and cut it in two and bring it over another strand to get it to move out and separate from the parent strand. So all of these things have, all these polymerases they call them, have been developed so that all this cell division which can occur, and again the DNA molecules are very long, can occur within 20 minutes or an hour sometimes. So everything goes very fast. Okay, I'd like to talk a little bit about another miracle that has occurred. All of this is pointing to the fact that we have a tremendous complex system, but it's a system that works very well. It's almost like there's some intelligence involved with developing a system rather than just a simple self-assembly. One of the things that everybody knows what the fish salmon is. Salmon has to convert some of its DNA into a sperm cell, of course, to fertilize the egg. And to do this, they somehow must take all of the coating off of a DNA molecule. And the coating on there is primarily histone. And we'll talk a little bit about histones, but the presence or absence of histones has something to do with the regulation of the genes that are coded on the DNA molecule. But anyway, to get it in, all of this DNA of a sperm, to get into a sperm cell that the salmon has, it must take all the histone off of the DNA, and then it puts in this sperm cell. And then after the sperm connects with the egg and it's you have a new individual, it returns all of this histone back to the DNA. So I mean, who could in advance predict that something like this would happen? It's really tremendous. And the histones are interesting in themselves, because I mentioned the pea plant. The pea has the histones that are very closely related to those in a cow. The reason I know is that you can buy histones in a commercial production, like in Sigma Company and Aldrich. And the ones that are in the pea system are almost exactly the same as the ones in the calf. So this happens more frequent than you would guess. And once there's the function determined in here, usually the function has mimicked some of the same DNA sequences that are in a lot of other organisms. So I want to mention one other protein. There's a protein called defensin. We found it in pea systems, and it's the gene that produces the defensin is activated when it's resisting a fungus. So we thought that it's probably somewhat unique, but then as we did more research and got it on GenBank and compared it with other proteins, the products of other genes, we found that this defensin is found in skins of frogs, it's found in scorpion toxin, and it's found in some of our own red blood cells. So even though the organisms that we're dealing with in terms of physical features and everything are quite different, the mechanisms for carrying out these reactions have a lot of similarities. One last example I want to have in terms of similarities is, if these are kind of sounding like Lego bricks, you know, that you can make them do a lot of different structure, make them look like different animals. The next one is ubiquitin. This is a protein that, as the word indicates, is almost in a lot of other organisms. It's everywhere, ubiquitin means. So I was wanting to work with determining if there's a ubiquitin in pea tissue, if we could pull it out. The animal people had already figured how to profile ubiquitin and had developed anisera toward it. So I thought, well, what's the chance of using animal ubiquitin anisera to pull out ubiquitin from pea tissue? So I went to my immunologist friend, and she said, yeah, it should work, because if they're that close, and it did indeed work. So again, another compound in animals the same as it was in plants. Now, I want to spend just a short period of biblical miracles and just say something about it in terms of the biochemistry involved. I especially like the biblical stories and the miracles, and they have been portrayed really good in a lot of stories in movies. And the biblical, the miracles that are supposed to be divinely determined are still occurring. When I was in France, I almost got the chance, if I'd had my connections right, to get to Lourdes, France. But in Lourdes, France, there's some fountains and things, at least I've heard of, where people go with their sickness or whether they're crippled or whatever, go to Lourdes, and a percentage of them apparently are healed. So some miracles which are divine related are still occurring. Now, the miracles that Moses did in terms of the burning bush, and what Jesus did in terms of healing leprosy, I mean, that's all within, it's not near as, what I would guess would be as complicated as setting up what was in an initial living cell. But some of the biblical miracles are suffering a little bit from not having the base of information that we have now to make our determinations. And so the authors of the Bible, when they talked about Jonah and the whale, and some of these things which are a little harder to fit into the biochemistry part. The other thing, when Jesus did the fishes and the barley loaves, when he fed the multitudes, this would, of course, in reality require all the kind of biochemistry it takes to make loaves of barley loaves, and all the ingredients you have in a fish. So biochemistry there was supernaturally developed. Some of the other miracles, like changing water to wine. Water is H2O, of course, and the ingredients of wine is ethyl alcohol. So ethyl alcohol is a very simple molecule. Of course, all the other components of wine would be very hard to come with, so it would require a divine miracle. And other biblical miracles, like Moses' parting of the waters, are rational, and the geologist probably could give you some rationale as to what kind of things would occur on Earth to cause the waters to part. Now, there seems to be some desire for people to see things like this in the movies, see where abnormal things, supernatural things are occurring, like Spider-Man, who defies gravity and all these sort of things. So our population needs to understand what biochemistry is and how it would have to be portrayed in anything that you would propose would be a miracle. And I guess my conclusion is that the ingenious of making a living cell is so tremendous that self-assembly is not going to suffice to develop something like that. So my conclusion is we got here somehow. We know that what we have here is real. It can be repeated. We know it's in the cells. We know it's in all the cells. So whoever, and if this is a divine establishment, it's one of the best that's on the earth, and it would compete with all of the biblical stories. So that's the end of my talk.